Okay, so welcome everyone. My name is Lourdes Figueroa. I'll be your host today. And today we'll have a presentation about DIY bio. Today we have Tom Peters, from a doctor from Erasmus Brussels University for Applied Sciences and Arts. And Tom got his PhD in biotechnology at KU Leuven in Belgium, where he elucidated signal transductions in yeast. So welcome everyone. Please remember to keep your mic off. You can make any questions through the chat. We'll have time at the end of the conference to solve them. So Doctor, I leave you with the speaker.
Okay, thank you. So I'm gonna give a talk. It's more, I will give some examples about DIY biology. I will show some graphs, but I will not show, I will not bore you with all those graphs and all that. So just that you have an idea about what DIY biology is about. Also, when I finished this presentation and I went through it again, and I finished it yesterday late, so sorry for that. But when I went through it, then I saw also like, okay, it is very Euro-centered. So, but that's mainly because this really started in the United States, and then it is follow in Europe, but it's getting global. So it's really is a global movement, which is important for you to know, and you can all participate in it. So in the front page, I also put something that you can scan right now, so you can become a LinkedIn buddy, or later on when they hand out the slide, that's no problem. So I got properly introduced, but here's a small introduction about who I am. So as you can see, I focused a lot on biotechnology and specifically on yeast biotechnology. And since 2009, I'm a teacher at Erasmus University College in Brussels. And in 2016, I started the Open Biolab. And then since 2017, I'm a scientist there. And now I am also an advisor, knowledge transfer. And I started an Open Bioincubator. And in the end, I will make some advertisement for that as well. So what is DIY bio? I will start with a definition, and then I will show you some more results and things that people are doing in that movement. So it's important to know it's a global movement, which is really important. So and it has a goal. It wants to spread biotechnology beyond the traditional academic and industry institutions. So what does this mean? That is, they want to make biotechnology accessible to everybody. If you saw the talk of Danielle just before mine, then you know we're building a device and we want to distribute this device so people can use it themselves. So really, biotechnology, we feel, should not only be something that's only confined to academic institutes or in industrial institutions. We think also everybody should be able to start working with it, and of course safely. So that this is also an important issue of this movement. So every year since 2000, I think it was 2017, in the MIT Media Lab, they started with the Global Bio Summit. If you really have the time or the opportunity wants to go there, you should really attend it, because then you see people from all over the world coming there and talking about what they're doing, spreading and sharing all these ideas. It's really amazing and very nice thing to go. And that's also where actually I met a student of Danielle. I started to talk to her, and then she gave me information of Daniel. We started to email. Then he flew over to Belgium and we wrote a proposal. And now we have a project running in Peru, which is really amazing. And it started over there. Also, I met the organizers there, and that's why I'm introduced and invited today. So this is DIY biology, a term, a controversial term I should talk about a little bit is biohacking, because maybe you heard about biohackers, and actually it is really a diverse term. So you should be cautious when you're using it. A lot of people in this movement call themselves biohackers. I don't call myself a biohacker. I am a biologist, so I call myself a biologist. And also because I think it's been used by different people in different means. So this is why I want to share a bit more about this term, what it means. So Elliot Roth, he wrote on his blog something about what biohacking is. So as you can see here, this definition. So it's about creating and using tools and methods, to modify biology for a novel purpose. And some people also use it to change the human condition. So here you have a nice graph, an overview. I used it last time in a presentation, and then the person who made this overview texted us and said, "Man, if you use my my slides, do include me and show a picture of me." So I did. It's not Wim Hof, but it's Amal Grafstra. So Amal, when you're seeing this, you're included. So you can see it's a very diverse definition or what biohackers are doing. You can see Wim Hof on one kind so of the slide. So he's a guy from Holland, and he's taking ice baths because it lowers his metabolism, and and he feels that by doing that his body and mind got better in touch, and his stress level reduces and all that. So this is one way of body hackers. So are people that are changing their habits in order to become more healthy. So under that you see the quantified self, and I think most of us are already working with quantified self beings without knowing it. So but if you ever went running and you're wearing a belt, that's keeping your heart rate, and you're watching it while you're running, you're quantifying yourself. That's when you're running and you see, okay, my heartbeat is going up, I'm gonna slow down a little bit, or maybe it's too low, I'm gonna do a bigger effort. That's already quantified self. So you're measuring your body, and you're changing your behavior with the goal to become more healthy. And then you see those two guys on the other side of the slide. So those are people with bionic implants, and Neil Harbison is a nice example. This guy is colorblind, and so he put a device on his head, as you can see, and it can scan wavelengths. So when he puts a color in front of it, the wavelength of the color will be changed into sound, and that will give a vibration into his ear. So, and that's why he says that right now he can even see or hear UV light. So, and he's even officially registered as a cyborg. And then you got Amal Graafstra, and he implanted, as you can see in the picture, so you see his hands, but it's photoshopped, of course. And then he implanted some chips, and he can open doors with it and all that. So, and he has a company about that. So where they implant chips in your hands, and you can, you can do that yourself also. But that's not what I call biohacking. So you see, biohacking, those guys also call themselves biohackers, and they have all right to call themselves biohackers. But you see, it's a diverse term. So I don't like to use the term biohacking when it comes to me. So when I talk about biohacking and what I will explain about, it's more about the DNA RNA hacking. As you can see underneath the biohacking, so they're in orange, yellow, you see DNA RNA hacking, and that is what I'm doing as well. One other person that's kind of in between is Josiah Zeiner. So I will talk about him a little bit about his institute, the Odin, what they're doing. So he also wants to make biotechnology accessible to all, and offer it at cheap prices. But he also at one time injected himself with the Cas9 so with CRISPR. So he put CRISPR on his body, and with the means of having increased muscle mass. So later on, he wrote in his blog also that he kind of regretted doing that. But you see, he's kind of in between, and I think right now he's more focusing on this DNA RNA hacking again, and he really wants to make biotechnology accessible to everybody. So that is the main goal. So for me and for a lot of biohackers, and I found this also on a website, and on my last slide, I will give you some overview about interesting things that you can read. So what is biohacking for most biohackers? What is it and what isn't it? So for most DIY biologists, Yes, we build GMOs. Yes, we do so, guilty, but not human genetic modification. We're not working with pathogens, so we play around with bacteria or with yeast, and we do genetically modify them, like if you saw in the previous talk. So we genetically altered the bacterium, but for the better. So it can do something that we can use. We're not going to make bio weapons, even if you would want to do that. It's hard, you know. Don't do it, you know. But it's also hard. We really want to make something that is helpful, something humane that people can benefit from. Also, like you saw in the previous talk, what we did, Daniel and I, we just were looking at what was already published and we rewired it. So we're not like in an academic institution unraveling a new pathway or something. We're using what's there already and we're building new stuff. And also, and this is very important and really in contrast to most academic institutes, we're working open source. And I will show you some examples of people that put even all their failures, and I think that's also very powerful, online and show what they're working on. So open source instead of closed. So that is what biohacking is for me, and I will not use that term anymore, okay? But so now you know. So in 2014, there was an article published by Gunther Zeufried, who is a DIY biologist from Austria, and he put down the characteristics. So again, you will see some things as in the previous slide. So you see it's interdisciplinary. So I will show you some examples. Some people, biologists, came together with engineers and they built new devices. This is a very interesting field. I really believe strongly in that, and I will show you some examples of that. Not for profit. Well, some people build things that they sell, of course, and that's perfectly fine. But it's just to make their lab kind of sustainable. It's not about the big money, you know, like not like big pharma. So primarily, so it says primarily not for profit. Also, they find cost-effective ways because most DIY biologists they just start a lab in their garage or wherever they find a place or even in their own sleeping room. So and they don't have a lot of means, so they work cost-effective. And then the very important thing again, the open source. Like you know, you work together, you share what you've done, and you continue on that, okay, and always acknowledge the others. And then it's about making it accessible to everybody. So that biotechnology, it's useful for all. It's not just something that a few people should use. It should be accessible to all. So how did this start? It's really interesting to know when this is movement start. And well, the first thing was already published in 2000, where they said after finalizing the human genome, there will be amateur genomicists that will explore their DNA. So that was just mentioned. So there were none yet, but people were already predicting, okay, this is going to happen. And in 2005, a professor, proved that he could build a lab in his garage. So he bought stuff, and in America, you can do it easily on Craigslist. You just buy old lab stuff. Even the Odin, you know, I will show their website later on. You can buy for really a few dollars. You can buy a PCR machine. So, and he built a working lab in his garage just to prove, okay, this is possible. And then you see this girl there in this small closet. It's I think 1 meter on 1 meter, and I think you guys use a metric system as well, so you know what I'm talking about. So, but she has this very small closet, and there she did laboratory work on a disease that her father was suffering from. And she wanted to know if she also had these genes. And also it shows, you know, you just need basic tools and you can do that at home. And then already early in the movement, DIYbio.org, a website still existing, started, which organizes everything that's going on the globe. So on DIYbio.org, you can find a lot of biolabs all over the world, or open community labs. If you're starting one yourself and you're not just on there, please go and put your lab on that website. And then in 2009, 2010, the first real open biolab started in New York City. Bacteria started an open source biological art platform. Then in Europe, a bit later, La Paillasse started in Paris by Thomas Landrin. And then in 2017, there was the first global biosummit. I think in the beginning were just a few hundred people, but now I think there were already more than 400 participants on the last one, and it was just the third or fourth time that they organize it. So if you now check diybio.org, you will see there's a lot of open biolabs or open community labs. So how did it start? Well, there's four things, actually, four things that happened around 2000. First of all, the expansion of synthetic biology. Unfortunately, I don't have the time to talk about this topic any longer than just this, but synthetic biology is a new discipline in biology. It's 20 years old, and they regard cells as an engineer. So, you know, a cell is built out of different compartments, and by rewiring all those compartments, we can build new cells and cells that behave just as we want them to behave. So that's basically, in a nutshell, synthetic biology, and a lot of people got inspired by this, by synthetic biology field. And an important other thing, on which I will show some slides later on, is a lower cost of technology. If you check, so be aware that this is a logarithmic scale. So you see, so the drop in cost it's really significantly. So you can see in the beginning that sequencing the human genome costed $100 million, and you can see right now you can do that for yeah, and that was even in 2009 for $48,000, which is just okay. You know, you can buy an expensive car, or you can just sequence someone's genome, you know. So it's getting really cheap. And you see the blue line, that was a trajectory. So it's really dropping, and it's getting so cheap because of new technology, and new technology that was invented so you could sequence for cheaper and cheap devices. Another thing, and this is what happened in the United States, and but you can see it all over the world. But here in the United States, it was really dramatically. Well, if you check, just don't check the dotted lines, but if you check the full lines, and if we see the blue line, we see that every year there's more and more PhD students. But if you check the orange line, we don't see there's a lot of positions in the end. So what do you have? A lot of people that pursue career in sciences, they have a PhD, but they cannot stay at the university because, you know, you have to wait till the professor drop dead. I'm not encouraging anyone to do something foolish, but you see that can, so that's just the thing. So there's not a lot of positions. You can go, of course, to work for companies, but you had a lot of people there, and they're a bit disgruntled, like, okay, with this diploma we want to do something useful, and they started to work or become DIY biologists, you know. So but they have the proper degree in that, and they started to organize themselves. So that's a fourth important thing. And I will show this example a bit later. They started to build a community, especially with this website already. They start to build a community and share. So a bit more about the lower costs. Really interesting here is the Bento Lab, and I put the prices also in Quetzal. I think I pronounce it hopefully correctly, so that you can see what it should cost for you. But you can see this is a lab. It has a centrifuge, gel electrophoresis, and a PCR for 1,200 euros, which is, and it's the size of a laptop. So Bento Lab, a really amazing device. They're also winning design prizes. It's really nice designed also. And here you see the nanopore of Minion, which is smaller and cheaper than an iPhone, and I think it will be more fun as well also. And you see Sebastian Kokioba, one of the most known biohackers. He used it to just sequence a plant, Oxalis stricta, at home. So he used his device. Another thing is here you see a standard PCR machine costs about 3,000 euros, but you also have the open PCR. So it's like an IKEA. I don't know if you have IKEA, but there you can buy, you know, these closets, and you don't have to put them all together yourself. So you can do that also here. You can build your own PCR machine, and it's open source. But then you can also do this, and this is really amazing. This is a group. It was published, and with some soup cans and some hacking, they built a PCR machine for $100 or €100. So just with a soup can and some wooden sticks and metal wire, they glued it together and they made a PCR. And they even took it to the next level. They had to put a little bit more money into this. I think this one costed 300, but you can see they even got a diagnostic paper with it. So so you can really use it. It's really cheap and you can use it. So when I saw this, I was really intrigued by it and said, okay, we're gonna do that ourselves in the Open Biolab. And this is what we did. So we built a PCR machine ourselves. And I can say we improved the Open PCR a little bit because you don't need a computer to um, to get this fired. And then a DIY gel electrophoresis system. We didn't use platinum, we just used iron. It oxidizes, but a paper clip costs nothing, so you can just discard it, which is waste, I know. But you can at least have a gel electrophoresis. But then I met Cristina Cipriano, and she made one from a Tic Tac box. This is a Tic Tac box. So this is really amazing what she done. You really should follow her. She does amazing stuff. And if you would like to do that yourself, there's several websites on which you can find some information about this. Um, I see I don't have to continue a bit faster, so we'll go through the slides. So what we done here, we built a PCR machine which is 3D printed, the red one, and then we had some renders because I think you can also make it fun, like the Metallica themed one. It's just an idea. We didn't build it in the end, but it should play Metallica songs all the time when it's operating. We also organized a course about this, Pimp My Incubator, for youngsters in Brussels. So people between 12 and 17 years old, 20 youngsters and some teachers came a week to the lab and they built a device themselves. I'm now currently working hard on getting new funding on organizing a second one and putting everything online because we had some problems with well we have everything but it's not there's some problems in the code, let's say. I'm not going to go into details, but we really would like to open it also that this is really accessible to everybody. We just don't want to keep it on our own, but I'm working on that. So if you check the community, I already told you, so a lot of those DIY biologists actually do have um, a degree in biology, and if you check them, what they mostly like to do, you see, first thing is education. They're really focused on education, on really putting it open, like this project with Danielle that Danielle just explained before me. It's also something that we want to use and give it to the people so they can use it and know more about it. And you can also see they're very much focused on transparency, like really we want to share everything. And also when we have our biosensors ready, then of course we want to publish it and that people can use it. And here there's a lot of work to do because this is something a survey in the United States, and here you could see that most scientists think that it's safe to eat GMOs, but the most people in the public don't. So how can we, so why, is the public opinion so against GMOs and scientists not? So there's a gap, and we also believe that we should make that gap smaller. And by making biotechnology accessible and so people understand it, we believe this gap will, or they will go towards the scientists. That's what we, at least what we hope. So, and it's a responsible community. So when they had a meeting, and this was already really early, there was a meeting once in Europe, and there was just a few people coming together. But one of the first thing they did, and also on the Global Biosummit, there's a lot of work put into that, is put a code of ethics. So this is maybe a little bit dated. They put one from Europe, one from North America. You can still find it on diybio.org. But also the Open Bio community is putting a lot of effort in writing a code of ethics that everybody can adhere to, can stick to, and so we want to make we want to make things that are beneficial, to make things better. And I think that is really important and really important to know. So, you know, those DIY biologists are not nutcases or crazy professors. You know, it's people that want to make biotechnology accessible for everybody. So here, the Odin, they were the first ones to offer a DIY bacterial gene engineering CRISPR kit for just about $150, which is super cheap. We bought one, but then it doubled the price to get it through customs. But then they also made one where you can genetically engineer frogs for $300, so which is super cheap. And you know, if you check this DIY bacterial gene kit, you even get a micropipette in there. So it's really everything what's put there on the picture. That's what you get. So you can instantly start experimenting yourself. Um, there's other companies as well. So Carolina is a very interesting company as well. They also have some kits, and you can see it's not for a lot of money that you can buy stuff there and use for education or for experimenting, of course. But those things are focused on education. Bioret, which is a renowned company that they sell a lot of biological tools to academic or research institutes, but they also have a part that focuses on education. And an interesting thing is a P-Glow bacterial transformation kit where you can make bacteria fluorescent. They sell it, and it's not a lot of money. And since recently, they also have a CRISPR kit, and I just figured out in the beginning they were only selling it to North America, but now already in Belgium. So I ordered one today. So we're gonna experiment with that also. 267 euros. That's not I mean that's something that we can that's not too expensive to start working um, with that device. And then a very nice company with whom we collaborate a little bit is Amino Bio, and they wrote this book, Zero to Genetic Engineering Hero, just $35. A nice book that explains on how you can start doing your own experiments. And they also have a chapter about safety and all that. Very interesting book, very cheap, I believe, and also it has nice protocols and things you can do with youngsters or yourself. And they also sell different kits. You can check on their website. There's a lot of things that they sell, but here you can see for just $410, you can buy that's a centrifuge and then some kits with whom you can with what you know that can help you to do a little bit of genetic engineering. So uh, you know, I'm flabbergasted. Just I would just buy it for the centrifuge already, you know. Um, we also started doing some stuff like that. Um, we made a let's swap kit, but we really should go to a new iteration um, where we put some stuff together for some schools in Brussels where they could make bacterial grow plates with common grocery and kitchen supplies. So it just we took some agar that you can buy in any food store, and then some yeast that you can buy in food store, and then some yeah, we call it bouillon, you know, some flavor salty things that you can put, you know, to pretend that you know how to cook. Those things we mix them together, and then we used an IKEA high pressure stove. So it was just it costed 60 euros, and with that you can sterilize the media. We pour it in jars, in a jam jar. We pour it, and you can grow bacteria. So, which is really nice. You can see here some experiments. We tried several bacteria, and they grow nice on that medium. And we built something for some schools and teachers to experiment with that. But we really have to make a new iteration. But that was our Let's Swap initiative. Something that we would like to continue with is to couple this with the agar art, which is something that's very nice to do with youngsters, and that's to make paintings with bacteria. So these are some examples that some of our students made um, in school. So, you know, just with pigmented bacteria, they made some drawings. There's always every year an agar art competition but it's also something nice to do in the classroom because you learn them how to make streaks, and then you tell them, okay, now bring it to the next level and draw something that you like. And these are some examples. We also this pic is with a Vibrio fischeri, which are luminescent bacteria that we use to cheer on our national soccer team that became third in the World Cup, something that everybody will forget, but we were happy that they were third. So some examples of community projects. I will just show you some examples about what people are doing in community labs. So here you have a counterculture lab and BioCurious, which is a fantastic name, BioCurious. They're open community labs at the west coast of the United States. They also participated in the iGEM competition, and they are working on making vegan cheese. So what they do is they want to genetically alter yeast cells, so the yeast cells will start making the cheese proteins, and you could make vegan milk. So there's no milk from, so it's cheese that's not made from milk from a cow or a goat. So it's just coming from yeast cells, which are genetically modified, and they make the milk proteins, and then you can have a fermentation, and you have real vegan cheese. I put also the website there, so you can always check it out or participate and help them out. Kombucha genomics, also something on food. Kombucha is yeah, it's a drink. It's a fermentation, and there's bacteria in it, and there's yeast in it, and it's not really known what's in there, and they want to characterize it to really see what organisms are growing inside of that. And as you can see, that's already a bit more labs involved. So BioCurious again, but there's also lab, Biomakers Lab in Peru, Bos Lab in Boston, Sound Biolab. So it's different labs collaborating again, you know, like again in this open atmosphere, working together on tackling this problem or this research question. Open insulin is one of the of those first more open community research programs that started, and I really like what the slogan underneath there. So it even became a foundation. It says healthcare is a human right, and they're damn right, it's a human right. So what you can see is that insulin is not accessible for everybody. Some poor people could not afford insulin, even in the United States. And there was a guy, and he said, "Okay, this is just bullshit. I'm just gonna build it myself." And a team of, yeah, okay, I put the term biohacker here, but it's a DIY biologist again, and they group together, and they're really working hard on making insulin together. And also it's not just working on making an insulin that's open to everyone, but also it's about creating awareness. So they also have educational programs. They're a foundation right now. They really got a lot of funding, which is fantastic for them, and they're really doing a great work. You can also check it out, openinsulin.org. You can see what they're doing. And something that which I also like is this the development of Sliberia, which is a generic version of Glibera, which used to be the most expensive drug in the world. So for treatment, you needed to pay $1 million to get treated. It was gene therapy, and those guys call themselves biohackers, and one of them was involved was Andreas Sturmer from Austria, who is really an awesome guy, and whenever you got an opportunity to see him speech, you have to go there. So Andreas Sturmer, um, so and they go together, and so with different people from all over the globe, they build an alternative, and they say, okay, they made a prototype. Okay, so they didn't go to clinical trials and all that, but they showed, look, you sell it for 1 million, but we can build it for $7,000. And it was really to show this big pharma, but like, look, you know, this is disruption. This is important, that those And okay, I understand those big pharma companies, they also put some money into it and also investments, but also they got also tax deductions and and PhDs that got funded by government. So let's be honest about that also. Um, and in Belgium we had so after that there was an even more expensive treatment, and in Belgium it was something because there was this girl suffering from a disease and it costed 2.4 million euros, and they got the money because a lot of Belgians donated money so they get this treatment. But this is ridiculous. We shouldn't allow this. Really funny also, guy Sebastian Kokioba, because what I really like about him is also he has a lab book. It's open, so it's an open lab book, and on his Facebook he puts everything that he's doing. And also one thing that I really liked is my failures and my successes. So because you can learn a lot from failures as well, and no one in academia is publishing a paper that says, "Look, we tried this and we failed in that," and that you're on. It's only a good no-show. And sometimes from those failures you can learn a lot, or at least you're not reproducing the same failures. So which is really interesting, and he's doing a lot of different things. um, and of course you can follow him on his Facebook, and there he posts almost every day what he's doing, and mostly it's on working on flower enhancement. So really interesting, also a very cool chap. And this is something recently, at least, I had to talk about COVID, of course. But this is also an open community initiative where Ellen Jorgensen, she founded the first open biolab in the world, Gene Space, but she's not involved there anymore. And she wanted to make an 1 hour COVID test. And she grouped together with people through Joggle, which is called Just One Giant Lab, and which is an open, well, and digital laboratory where people get together and they have an idea and they want to work on something, and there they share their results. It's a communal working space for scientists or people that are doing some stuff in science. And she posted on there, so you can read everything they're doing. You can get involved. There's several things going on Juggle. Interesting to check it out. And it's open community. And here she just won a prize with her design, because they proved that they could create a COVID test that shows the results within 1 hour, which is really interesting. Um, I'd like to finish with something that I thought was also funny. What a DIY biologist did, but he also has a degree in biology. Sascha Karberg in Germany. So where he was living, he saw that there was a dog leaving some unpleasant treats in front of his door, and he wanted to know which dog is doing that. So in his neighborhood, he was throwing tennis balls to the dogs, and the dog picks it, gives it back, and he was collecting their saliva, so the DNA of the dogs. And then he was comparing it with the little package that was always in front of his door, and then he found the dog responsible for that, and then they went to the owner to confront him. So this is also something you can do in your DIY biology lab. I really love this. It's something funny, but it shows the power also of DIY biology. You could do this. It takes a lot of time, but you could do this, and it's not a lot of money. You can do this. So um, if you're interested in what we're doing, I would ask you to follow our lab, the Open Biolab Brussels, on social media. You can always send us an email. Please just send us an email to our regular email and not via Facebook or Instagram. Um, but on Facebook we put some stuff about what we're doing, and on especially on the Instagram, all the stuff that's going on right now. Not a lot because due to COVID we're closed, but we're developing a lot of workshops right now, and we will publish that later on. Also, something I would like to make a little bit advertisement for is the Open Bio Incubator, which I just started. So the Open Bio Lab, that's really focused on tinkering with biology, which is really for high schoolers and for schools and teachers and people that want to start with DIY biology. But the Open Bio Incubator is for people that would just like to start a biotechnology business. So we have a lab which is equipped with proper lab gear, so not the IKEA stuff, so that we have in the Open Bio Lab. We've got another lab, the Open Bio Incubator, and there you can start putting your ID into a proof of concept, which is the goal to help people that have an ID in biotechnology, but they of course don't have the means to build a lab. They can start in an Open Bio Incubator. They rent the space. It's a shared space. You can use all the devices, and then hopefully your ID will accelerate and you can move on. And we already successfully at a company that's going to leave us because they got their first results in the Open Bio Incubator and they got some investment money and they're continuing. So we're very proud of that. And to end, um, here are some stuff that I read to build this presentation, and it's up to you to check that as well. I think the organization will share my slides, or they can share my slides, and then you have these things. So thanks for listening to my talk. If there's any questions, I'm happy to address.
Thank you very much, Doctor, for this excellent presentation. So please, attendees, if you have any questions, you can type it on the chat and we'll read it to the doctor. So let's wait a bit to see if anyone has.
Yeah, thank you very much, Tom. I have a question. So when you started doing like this bio hacking stuff, are you doing it at your home or at your lab?
No,
Or
at the lab.
at your
One
closet?
time, yeah, no, one time I was building a small lab in the garage, so to do some bio art stuff, mostly. But no, because we built a lab in the school, so I'm not doing it at home. It's more convenient to do it in a lab at a school, I have to admit. Um, but I think about making a small one for bio art, some stuff that I want to explore on the side on my own, which I can't do in that lab. Yeah.
And do you think this like biohacking can do it like through video conference, like Zoom or something like that, that we can accomplish here in Guatemala? You can teach us how to do
Yeah,
biohackers?
it's a lot of people are already doing that. If you check just recently, but it was a bio art thing by so the return of Dillman, which is a CRISPR experiment in vitro, and it was done through a video conference, and people in the lab were doing the same steps also. So yeah, this is possible. You can do that for instructions. Yeah, yeah. But what we would like to do is to, like Danielle also said, we would like to build a website where we tape ourselves beforehand and put, yeah, the movies online instead of doing it live. But maybe, yeah, you know, when this epidemic stays for a long time, then that's really something to consider. Yes.
Thank you. And what are the challenges to be a biohacker? It's like it's regulated or it's not regulated. It's against the law to have a lab in your home or not.
No, yeah, it's that's the regulation is something fishy, but as long as you're not working with pathogens, and if you alter an E. coli K12 that cannot spread in the environment, it's fairly safe, of course, to do that. But I really encourage people to do it in an open biolab, and that's also something hard to start such a lab and to maintain it, especially if you do it only with volunteers and limited funding. That's quite something. So you really need motivated people if you want to start um, your own open biolab. So I was in that sense a bit lucky that I have my institute behind me. So we had a laboratory facility and we started with the workshops. We could start with the workshops, and we didn't have to invest in the space. So I do realize that yeah, we're lucky in that sense. So that's hard for community labs to first get the space, then get all the equipment. Yeah, I think that's hardest for them.
Yeah, thank you. I think most of the people here are students and part of universities. So I think we can start from the university, like ask for
Yeah.
help with that sport. And let you know that in the chat, here is Anje. He's from Countercultural Labs, and he's happy that you will notice about the lab, and shout out about the vegan cheese and kombucha Yunami.
Ah, yeah. Yeah, they're doing amazing stuff. So yeah, and they're a known lab. I could not talk about every lab I told you in the beginning, but these are nice examples, and you can follow them. They're doing amazing stuff. Yeah.
Yeah, thank you very much. I don't know if anyone has any more questions. Ana Fortuna says, like, thank you for sharing all these DiBio project themes and excellent presentation.
Yeah, thank you. Thank you.
And I will let you
Also,
with
I have
Lourdes
a
again.
question. Okay, first I have a question. Do you ship worldwide those little packs for making the art with egg art?
Yeah, we don't ship ourselves, but there's so yeah, because where we bought them from, we bought them from an institute, and we specifically had to sign a document in which we said that we don't share the strains. But what I can do, if you're interested in that, I can, but then you have to send me an email or through the Open Biolab. I can send you a list of organisms that we know that work. Organisms, because with the pigmented bacteria, sometimes those bacteria don't want to produce the pigment. So and something that and I don't have any stocks there, but with AminoBio, which is interesting, is that they sell the plasmids with the pigments, and then you put them in the E. coli. So and it's not for a lot of money. So and then you will have a more stable expression of the pigment. So I encourage you to go through to them, and then you know it will work, and it's legal and all that, and there's no problem of shipping it also, because also if I have to ship it to you, I think it can be blocked at customs.
Yes,
So
it's complicated with customs. I think this is our
yeah,
real problem as Latin America region, because we have really big problem with custom, because they don't understand if the like the paper
yeah,
says plasmid, they say like, oh my God, this is bioterrorism. So
yeah, yeah,
this is
yeah,
like
yeah.
one of the challenge here, but I think we are just start moving forward that kind of project that everyone can, pues, it's just the bacteria that are not dangerous for anyone.
No, I know. Yeah, for the plasmids, or what you can do also, if you get a sequence, but not a lot of people, well, AminoBio will not share the sequence because they're selling the plasmids. Then you could, of course, buy G blocks. Then you just buy, you construct a plasmid, and you buy it from IDT or something. That's something that you could do. And also, which is interesting, is also to always check AdGene, the website, because you can order plasmids through AdGene also. So
Okay, sounds good. Thank you very
yeah,
much.
if you want to have more information, you can contact me. I'm happy to help.
Okay, perfect. I'll be sending you an email.
Okay,
Okay,
cool.
so we don't have any more questions on the chat. Just congratulations for the excellent presentation. Thank you very much. It was very interesting. And it's also encouraging to see that you can do it at home, kind of. So thank you
Yeah,
very
yeah.
much to all the participants. Thank you for being here with us today. We still have more conferences if you want to be on them. So thank you, Doctor, for this excellent presentation, and have a nice day, everyone.
Yeah, thank you, and thanks for the invite. Bye.
See you.
Bye.
Bye.